moving on to the last question of this chapter, that is question number 14. Briefly describe the following terms such as transcription, polymorphism, translation and bioinformatics. So we will be discussing each and every term, or we will be explaining briefly, describing briefly each and every term on the screen one by one. First we are taking the transcription. So we have to describe transcription. First of all, what is transcription? Transcription is a process by which RNA is synthesized from DNA. So simply the transcription is a process in which RNA is synthesized from DNA. Transcription is a three-step process. What are these three steps? Steps are included in the process of transcription, that is initiation, elongation and termination. In the process or in the step of initiation, what happens? The RNA polymerase enzyme binds to the specific sequence of DNA which is known as promoter and provides the single stranded template strand of the DNA during the process of transcription. Now in the process or in the step of elongation, this RNA polymerase enzyme reads the template strand of DNA and adds bases to elongate the chain. In the last step of transcription, such as termination, specific terminating sequence marks the end of transcription process. So this is how RNA is going to be made from DNA, and the process of formation of RNA from DNA is termed as transcription. So let's write answer number 14, and we'll be writing part A of this, that is transcription. Transcription. So what is transcription? It is a process of formation of RNA from DNA. Simply it is a process of formation of RNA from DNA and we call it as transcription. It is a three-step process. It is a three-step process. What are these three processes included in the transcription? That is initiation, elongation and termination. So first step of this transcription process is initiation. In this step, what happens? RNA polymerase enzyme binds to the promoter and providing binds to the promoter, which is providing the single stranded template during transcription. The next step of this process, transcription, is elongation. Now in this step, RNA polymerase enzyme reads the template strand and adds the basis to elongate the chain. Then the third last step of this process transcription we have is termination, in which a specific terminating sequence marks the end of this process, marks the end of this transcription process. So this is the description of part 1 or part A of this question number 14. We have described the transcription process, which is a 3-step process. We have covered all 3 steps of this process. This is the complete solution of part A of question number 14. Now let's discuss the B part of this question, that is we have to describe about polymorphism. What is polymorphism? Polymorphism is a kind of genetic variation. Polymorphism is a kind of genetic variation which leads to the formation of several different kinds of individuals among the members of one particular species. What is polymorphism? Polymorphism is a kind of genetic variation. It is nothing, it is a form of genetic variation which results into the formation of several different kind of individuals among the members of one particular species. Now, the varying sequences of nucleotides are located on the different positions of DNA helix. The varying sequences of nucleotides are located on the different positions in a DNA helix, which result into the variation and polymorphism in a population. This polymorphism is responsible for speciation and evolution as well. So let's write the B part of this question. What is polymorphism? Polymorphism is a form or kind of genetic variation which leads to the formation of several different types of individuals, among the members of one particular species. Now this varying sequence of nucleotide are located on different positions in a DNA helix, which results into variation and polymorphism in population. This polymorphism also responsible is also responsible for speciation and evolution. So this is the complete description of polymorphism, and with this we have completed the B part of question number 14, and moving on to the next part, that is C part, in which we have to describe about translation. So now let's discuss translation. What is translation? It is also a process. Now, translation is also a process in which the protein is going to be synthesized from RNA. Translation is basically a process through which protein is synthesized from RNA. Now, this translation is also a 3-step process. What are these 3 steps? Initiation, elongation and termination. just similar to the transcription process. Here also in translation we have 3 steps such as initiation, elongation and termination. In the process of initiation in translation, what happens? The ribosome assembles or the ribosome attaches to mRNA which has to be read, and the first tRNA will be carrying the amino acid such as methionine. Then in the next step, such as elongation in translation, what happens? The amino acid chain is being elongated. Then in the last step of translation, that is termination, the synthesized polypeptide chain is being released. So these all are the 3 steps included in the process of translation, which we are going to write on the screen now. Translation. It is a process of formation of protein from RNA. So simply translation is a process from which we will synthesize protein from RNA. It is also 3-step process. The very first step of this translation is initiation, in which the ribosomes are attached to the mRNA and has to be read. And the first tRNA in this process will be carrying the amino acid methionine. This is the first amino acid which will be carried by the first tRNA in the process of initiation of translation. Then the next step under this process we have is elongation. Now what is elongation? In the step, the chain of amino acids is being elongated by the addition of several amino acids. It is being elongated, the chain of amino acids is being elongated by the addition of several kinds of amino acids. The last step we have is termination. In this process, the synthesized polypeptide chain is being released. So in the process or in the last step of translation, that is termination, the newly synthesized polypeptide chain is being released. So this is the complete description of a process translation. With this, we have completed the third part or C part of this question. Now we will be discussing the last part of this last question, that is description of bioinformatics. So what is bioinformatics? It is the use of information technologies. Bioinformatics is the use of information technologies for collecting and analyzing biological complex data such as genetic codes and protein sequences. There has been an advancement in the field of molecular biology. There is the invention of computers, scientific techniques and instrumentations at the level of nano level. So these have helped in the development and progress of bioinformatics. So let's write the last part of this question. Bioinformatics. So bioinformatics is the use of information technologies. Information technologies. Bioinformatics is the use of information technologies for collecting and analyzing the complex biological data, the complex biological data and etc. with the help of databases, Now there has been an advancement, in the field of molecular biology. There is invention of computers, scientific techniques and instrumentations at nano level. These have helped, all these have helped in the progress and development of bioinformatics. So this is the complete solution of D part of this question, and with this we have completed all the questions included in this chapter. This is all for today. Thank you and have a nice day.